Hello friends, welcome back to episode 10 of Genomics for All. And today's topic friends, I'm sure everyone, all of you will be very interested and curious about how this is. And this is longevity, aging, how we can live longer, how to become immortal. These are questions that everybody has. And today's topic, I'll be discussing genomics for longevity or genomics for aging. This curiosity about living longer has been there from the ages. In our Mahabharata, as you saw, the sage Vishwamitra being immortal. Or in today's age, Brian Johnson, one of the eminent businessmen, he is spending millions of dollars to reverse aging to become, to increase the longevity to 150 years. That's the goal. So a lot of things in between have come, a lot of science has developed, how we can reverse aging, how we can live longer. So that is the topic today we will be discussing on how genomics can affect longevity, how genomics can affect aging. What is aging all about? We have one thing called the chronological age, wherein these are calculated in numbers. And the next we have the biological age or the cellular age as we call it today. That is the actual state of the cell or state of the body. Now, how does the state change from the chronological age is due to many factors, and this could be due to mitochondrial dysfunction, oxidation, free radicals, for example, they accumulate in the cell, and then DNA damage, and that is why genomics becomes very important to all of this. What we have seen, and all of you would have heard, is that technologies like calorie restriction, what people have, doctors and nutritionists and dietitians have evolved, is one of the tools that can help increase longevity or reverse or decrease aging. So calorie restriction, which in scientific terms increases autophagy, which is the cell's own way of degrading the accumulated free radicals or oxidative damage, that won the Nobel Prize in 2016 to Yoshimi. So that these are some terms that all of us would have heard, that calorie restriction, autophagy, this can actually help in increasing our longevity or reversing our aging. But what does genomics do to all this? That is the topic of discussion today. Do we have signatures or biomarkers that can tell us whether a person will live longer or not? The science is not yet very developed, but there are some genes like Klotho and TERT, which is a telomerase reverse transcriptase. These are markers that are being studied very thoroughly, because when scientists have done analysis of very long-lived men and women, these markers were different among them when compared to short-lived men and women. So very interesting studies are happening in this area of genomics for aging or genomics for longevity. And then within this genomics, I will talk about a subgroup of genomics called the epigenomics. Now, what is epigenomics? Epi is above, genomics. So anything that we are born with is the genomic landscape, and changes that happen in the genome belong to this category of epigenomics. Now, all of you would have heard methylations. So DNA methylations belong to this category of epigenomics. And here again there are two categories. One is the chromatin modifications, wherein histones, that is the chromatin part, the protein part attached to the DNA, can have modifications like acetylation, methylation, phosphorylation, sumoylation, ubiquitination, ADP ribosylation, etc. So now all of these changes happen to the histones. And now there is another category wherein methylations in the DNA itself, wherein CG residues are there. The methylation on the C determines the epigenomic landscape or the epigenetic landscape as we call it. Now these are methylations that are dynamics, which means these methylations are there and one can reverse them. So these are the ways scientists are going forward, that can we now reverse aging? Can we increase longevity by tackling these methylations? Studies are still underway. We are not there yet, but very interesting studies in the last decade have come that clearly define this epigenomic landscape and the epigenetic landscape that define aging and this is actually called now the epigenetic age, or the cellular age as I called it, and that is the number of methylations that a person has in a given time. So one can actually now intervene, play with these methylations and actually reverse aging. That is where we have come. What at my biome we do is we have a panel called the methylation panel or the epigenomic panel. What it does is it carries a 123 million basis genomic region which covers 4 million CPGs which are methylated in the human genome. So it covers the entire methylated part of the human genome or the epigenome. So we analyze that and we tell a person whether out of 4 million, your 2 million are methylated and 2 million are not methylated, just for an example, just for perspective, and then calculate the epigenetic age of an individual depending on where he or she is in terms of methylation. So this is the way forward. Very interesting things coming in genomics or epigenomics for longevity and aging, and very soon we'll see some very strong science emerge out of it, and maybe the day is not far where we will be able to intervene and affect our longevity and aging by different supplements, calorie restriction, autophagy, and many medications also people are talking about which could reverse aging. Thank you all for supporting Genomics for All.